Okay, so we're talking about spontaneous mutations, and modified bases are the major or main reason for those mutations to happen. The most common one is called a 5' methylcytosine spontaneous mutation. So this occurs when a methyl group is added to a cytosine base. Okay, that changes how the sequence is read and addressed. Once again, it's a 5' methylcytosine spontaneous mutation. So when that kind of mutation occurs, it produces a GU and GT mutation. And it also changes its compatible pair base, right? So that allows suddenly to the G to base with to be paired with either a U or a T. Okay, so without going into what a methyl group is and how it changes exactly, just remember that it's possible when a methyl group is added to a C, it allows suddenly for a G to connect with a U or with a T. Okay? This is what we would call a mismatch. Okay? The cell has mechanisms that are able to identify mismatch and repair them, but some of them slip through the system, so to speak, and become what we call permanent mutations, right? So here's the thing. The chance of a GU being repaired is greater than the chance of a GT being repaired. The system is able to recognize GU a lot easier, okay? And that means that this mutation that we call the 5' methylcytosine spontaneous mutation is usually not repaired, meaning that most of them result in a GT pairing. Okay? So you're referred to a movie, enjoy it. So when we are repairing a base pair that is incorrect, we can address that as a process called base flipping. Right? Okay, so here's the thing. We have a sequence, an original sequence, that is supposed to be perfect, right? And now a mistake has happened. Mistake meaning a mutation. If it's not repaired before the next replication of the molecule, we're in trouble. Why? Because it's going to be replicated with the mistake. Let's say that you sent to your friend a document, a Word document, right? And there's mistakes in it. And you don't want those mistakes to be, distributed to the world, right? You have to be able to contact that friend, tell him or her or them to not use that document and to not distribute it. But if they already sent it, meaning duplications have been sent, we're probably too late, right? So any mutation that we want to repair, we have to repair it before that molecule with the mutation, with the mistake, is duplicated. Okay? Does that make sense? So basically, any repair has to be done within one cycle of replication. Okay. Okay, so we find that 5' methylcytosine spontaneous mutation accounts only for 1% of mistakes in base pairs, but it accounts for 30% of the mutations. Okay, so out of all options, it's 1%, but when we look at the mutations, that's the most dominant one, and it accounts for about 30%. If we were to take a cell and deactivate its mutation, the canceling or the repair system mechanisms for mutations, we would probably have at least 3 times more mutations. Now, as we said, and I'm going over this again and again, it might result in an advantage, right? Or a disadvantage, or no effect. But we also already have learned that most of the mutations have negative effects rather than positive. So if I had to bet, I wouldn't do that if I were you, so to speak. The fact that uracil is not used in DNA makes DNA a lot more stable. Okay, and thus we find less mismatches in DNA than we find in our RNA, for instance. And also, it makes the backbone of the molecule a lot weaker than when we use thymine. So we've been talking until now about a hypothesis that one gene produces one protein, okay, and a very large family of proteins are enzymes. So enzymes are a subfamily of proteins. So there's the theory that is called one gene, one enzyme hypothesis, right? So let's say, when thinking about the subfamily of proteins that's called enzymes, what might happen if we have a mutation in a gene that's supposed to produce not just any ordinary protein, but an enzyme? This is where I'll remind you, hopefully I'm reminding you, that enzymes are kind of chemical supermans. And in order to receive the title of enzyme, you have to be able to catalyze and make a reaction speeded up a million times more than it would happen without the enzyme. So these are really super molecules, right, in a way. So the weakest and slowest enzyme takes a process that would take you 1,000,000 seconds and makes it happen in 1 second. That means that something that would take roughly 11.5 days is now taking you a second, and that's the slowest enzyme we're talking about, which is awesome, right? So now we have a mutation in a gene that is supposed to produce an enzyme. What are the implications of this? One, the enzyme becomes inactive. So the whole reaction that depends on that enzyme is canceled, and metabolism is affected, right? The second thing is an enzyme is activated, so the reaction occurs and metabolism is affected. When I say affected, I didn't say if it's an advantage or not, right? And another thing is that the enzyme is not affected, so metabolism, any effect to metabolism is not noticed. And by the way, this is rare to non-existent. So when a mutation occurs in a gene that produces a protein that is an enzyme, most likely metabolism function is going to be affected. So we don't want that, usually. Enzymes usually have subunits, right, that are either the same or different in shape, content, and function. And that is why the hypothesis that we started with has been fine-tuned, so to speak. So, from the understanding that one gene means one protein, and thus if the gene codes for an enzyme, one gene means one enzyme, suddenly we understand that one gene can produce more than one protein. Okay? If the subunits of an enzyme are identical, we're going to say that that protein is called a homomultimer, meaning that the enzyme has more than one subunit, but it is identical. Once again, when you have a homomultimer, you have a protein enzyme that has more than one subunit, and they are all identical. Okay? If the subunits are different, then the protein is called a heteromultimer. Once again, you have a heteromultimer, you have a protein, an enzyme that has subunits, but they are not identical. Okay? Okay, so the fact that we have heteromultimers means that more than one gene may be needed to produce an enzyme, or what we will call a protein-based complex. So every gene may code for a certain subunits. Okay, so now what we're going to say is, according to what we've just learned, is that one gene produces one polypeptide chain. Okay? And that polypeptide chain may be the whole protein or a subunit of a bigger complex. But it also means that if you have a problem in one gene, you'll have a problem in one subunit, and that might cause the loss of function, which is most likely, or an improved function, right? So we understand that. We find that there are non-protein coding genes, that what they produce is components of different apparatuses used in the cell, and take part in what we call the gene expression process. Now, this whole course was brought into the curriculum, so at the end of the day, or at the end of the course, you know everything there is to know, or a lot to know about gene expression and exercise. So this course is actually dedicated to gene expression. All right, how is the information in the gene expressed? What is actually expressed? What is actually executed in the transition between instructions, meaning DNA, to the mediator called mRNA, to the outcome, the product called called proteins, what happened? Did it even happen? Did it happen accurately or not? Gene expression. Okay, we're going to stop here and continue in the next video.